Good morning, everybody, and welcome to the fourth day of the Physiological Society meeting of 2021. And my name is Daniel Brayson. I am the early career trustee of the Physiological Society. And it's my great pleasure today to introduce the speaker, presenter of this year's Bayliss Starling Prize Lecture. And today we've got Samira Lakhal-Littleton giving her talk on local iron homeostasis, the mechanisms, importance and implications in honor of the Bayliss Starling Prize Lecture. And if I just say a few words about the Bayliss Starling Prize Lecture, it was introduced in 1960 to commemorate and honor the memory of William Bayliss and Ernest Starling, who played really seminal and key roles in identifying secretin, which is a gastrointestinal hormone. And importantly, they coined the term hormone. So effectively, really developing and inventing a new field that has really taken off and now is one of the bedrock subdisciplines of physiology. And so, as I said, it's my great pleasure to introduce Samira as this year's awardee. I'm very much looking forward to her talk. And if I just give you a bit of background about Samira. So Samira did her degree in human genetics at University College London before joining the University of Oxford in 2004 as a DPhil student in the lab of Vincenzo Sarandolo to investigate the role of amino acid metabolism in immune responses and tumor immune surveillance. And during this time, she became interested in iron regulation and went on to develop her career in iron regulation under the tutelage and supervision of Professor Sir Peter Ratcliffe at University of Oxford, since when she has done, well, I've just been looking at some of her papers, really seminal work in the area of iron homeostasis in a number of different tissue beds. And so really her work is excellent, and I think really is a fitting recipient of this year's William Bayliss Starling Prize Lecture, since it really crosses those disciplines that Bayliss and Starling were interested in, chemistry and physiology. So without further ado, I'd like to give the stage over to Samira, and I'm really looking forward to your talk, Samira. Please take it away.
Dan, thank you very much for that introduction. Good morning, everyone. I'm honored to be this year's recipient of the Physiological Society's Bayliss Starling Prize Lecture. I hope to use this lecture to give you an overview of the journey of discovery that my team and I have been on over the past 8 years or so. This journey has led us to understand the mechanisms, importance, and implications of local iron homeostasis. The mention of iron to most people conjures up the idea of hemoglobin and how iron is important for the transport of oxygen in red blood cell. Today's talk isn't about that at all. It is about a much more fundamental and wide-reaching role for iron in biology. To begin to appreciate that role, it's very important to take the long view of iron in biology. And I'm going to give you a very long view. In fact, I'm going to go back to the beginning of our planet. So it is well recognized that when our planet formed 4 billion years ago, it was essentially an oxygen-free environment. But then it went on to become a fully oxygenated environment. There were indeed 2 major oxygenation events, one about 2.4 billion years ago, and another more recent one about 500 million years ago. Before these oxygenation events, iron was freely bioavailable in the ocean and highly accessible. But the oxygenation events meant that iron was oxidized and precipitated. This is a process called banded iron formation. And so the advent of oxygen caused a major reduction in the bioavailability of iron in the environment. At the same time, there was an explosion in biomass. And this explosion in biomass came with an increased demand for redox active metals that can act or operate in a wide range of redox potentials. What this essentially means is, over the past few 100 million years of evolution, there has been a major decrease in the bioavailability of iron, accompanied by a drastic increase in the requirement for iron. Now, when you put these two things together, it is perhaps not surprising that many of the pathways that have evolved to acquire iron have had to do so under very tightly controlled conditions. Now, it is not surprising that evolution has chosen the iron pair to fuel the biology of life, essentially. The redox potential of this pair is modulated by the complexing ligand. And because of this pair, the redox potential can vary widely. And that means that it can operate across a wide pH range. And indeed, iron cofactors span almost the entire biological range of redox potentials in biology. So this pair is a very special pair, unlike any other redox metal on Earth. And so these special properties explains why this is the redox pair that fuels the chemistry of life. Many of the proteins that catalyze important reactions use iron. Proteins involved in the transport of oxygen, hemoglobin, for instance, in the storage of oxygen, myoglobin, the activation of molecular oxygen, reduction of ribonucleotides, electron transfer, redox signaling in heme groups of cytochromes and in iron-sulfur groups of enzymes such as nitrogenases. Now, these same unique properties of iron underpin its undesirable role in biology, and that of being a major source of oxidative stress. Now, it is well known that free iron causes the generation of reactive oxygen species by multiple pathways, including perhaps most famously the Fenton type reaction shown here. The hydroxyl radicals produced by free iron are very, very dangerous entities because they can react with virtually every biomolecule known to man. And in doing so, they cause irreversible damage to DNA, lipids, and proteins. And this damage underpins cell aging and cell death. With this in mind, this dual role in the chemistry of life and in oxidative stress, as I said earlier, it is perhaps not surprising that we have evolved pathways to acquire, store, mobilize, and use iron at the same time that we have evolved pathways to control labile iron levels and minimize the oxidative stress that comes from free iron. And this balancing act is at the heart of iron homeostasis. And in today's lecture, I'm going to tell you about our discoveries over the past 5 years or so, how the local hepcidin ferroportin axis is essential for this balancing act of iron homeostasis. Now, the key word in that title is local. Before I tell you about local iron homeostasis, I need to give you the wider context of systemic iron homeostasis. Now, systemic iron homeostasis is concerned with the supply of iron to the plasma to meet the demands of other tissues. We tend to think of systemic iron homeostasis in terms of supply and demand, with blood being the conveyor of both. So, as I mentioned earlier, free iron is a dangerous species, and as such, we find iron in the blood transported by a chaperone. This one is called transferrin. Most of the iron in the blood is used for the production of red blood cells in the bone marrow. Other tissues also require iron, most notably the cardiac muscle, the skeletal muscle, the brain. And while demand for iron in these tissues is far lower in magnitude than it is in the bone marrow, it is nonetheless very important for the healthy functioning of those tissues. Now, where does this iron come from? We traditionally think of our diet as the main source of iron. In fact, most of the iron that we use on a daily basis comes from the recycling of senescent red blood cells. This is a process that is carried out by reticuloendothelial macrophages in the spleen. They gobble up dying red blood cells and they spit out the iron again. When iron demand is particularly high, we tend to also resort to our liver iron stores, for instance, in growth or during pregnancy. Now, you will have noted that there is a common denominator required for the acquisition of iron from the spleen, the diet, and the liver, and that is the iron exporter protein ferroportin, or FPN. So ferroportin is the only iron exporting protein we know of in mammals, and that is perhaps very important to understanding the essential function of ferroportin in biology. Now, when transferrin saturation in the blood rises above a certain levels, the liver produces a hormone peptide called hepcidin. Now, hepcidin is the ferroportin antagonist. It binds ferroportin, causes it to be internalized and degraded, and in doing so inhibits iron export into the plasma and lowers transferrin saturation, so restores normal transferrin saturation levels. So this was a brief overview of systemic iron homeostasis. Now, what happens to iron in tissues once it is taken up? So this is local iron homeostasis. So I will tell you about the consensus, at least until recently, of how local iron homeostasis operates. If you look at any given cell, most of the iron in the cell is present in two forms, either within the mitochondria and the mitochondrial enzymes, or in ferritin. Ferritin is the protein within cells that stores iron and keeps it from reacting with other molecules. A very small fraction of iron is also found free in the plasma, and this is also required for various chemical reactions. Now, local iron homeostasis hinges on two processes, and that is the regulation of uptake and the regulation of storage. So tissues respond to iron deficiency by increasing uptake, that is raising the expression of transferrin receptor, and decreasing storage, that is breaking down ferritin to release stored iron into the cell. Conversely, tissues respond to excess iron by decreasing uptake, that is decreasing the expression of transferrin receptor, and increasing levels of ferritin to increase the scavenging activity of ferritin and the storage of ferritin in the cell. Now, the work that we've carried out over the past few years have given us new understanding of local iron homeostasis. What we have essentially been able to do is discover a third and non-redundant component of local iron homeostasis, and that is iron export. And it turns out that ferroportin is not just present in the tissues that supply iron, liver, the spleen, and the gut. It is also widely present in a number of other tissues, such as the heart, the brain, the kidney, and so on. In those tissues, ferroportin is not concerned with the supply of iron into the plasma. It does an entirely different function, and that is to control intracellular iron levels. Furthermore, some of these ferroportin-expressing tissues also produce their own hepcidin, and in doing so, are able to regulate this iron export cell autonomously. And so it appears that some tissues have a third mechanism of local iron control above and beyond uptake and storage. And this third mechanism is concerned with the control of intracellular iron level via export or regulated export. So where does regulated iron export operate? We found that regulated iron export operates in cardiac myocytes, in the pulmonary artery smooth muscle cells, in fetal hepatocytes, and in renal tubules. And I will take you through each one of these briefly. Before doing that, I just want to point out the challenges that come with the study of local iron homeostasis. Now, I remember very well first suspecting a role for hepcidin and ferroportin in local iron homeostasis about 10 years ago. This was a Friday evening experiment where I noted not insignificant expression of ferroportin in the heart, in the mouse heart, and also of hepcidin, and also noted there was an inverse relationship between the levels of the hepcidin transcript and levels of the ferroportin protein in the mouse heart. But before I could pursue the idea of local iron homeostasis, I had to see what animal models were available. Unfortunately, the animal models that were available at the time weren't suitable, because there were animal models of a ubiquitous deletion of ferroportin. And obviously these presented with embryonic lethality, perhaps not surprisingly, because iron is required for intrauterine growth. There were also models of ubiquitous hepcidin deletion, and these models developed systemic iron overload. Again, these models weren't suitable for the study of local iron homeostasis because it was difficult to discern whether changes in tissue iron levels were a result of locally operating mechanisms or simply a reflection of increased plasma iron availability. And so I decided that I needed a different approach to the study of local iron homeostasis, a painstaking one, in fact. So I decided that in order to understand local iron homeostasis, there were three interventions that I needed to be able to do. I needed to be able to delete ferroportin, I needed to be able to delete hepcidin, but also, and perhaps this is my favorite model, able to replace ferroportin with a ferroportin that was resistant to hepcidin. That is a ferroportin that continued to export iron and cannot be inhibited by hepcidin. And so we generated three conditional models, mouse models, where we have a floxed ferroportin, a floxed hepcidin, or a construct that can allow the knocking of a hepcidin-resistant ferroportin. That is the replacement of endogenous ferroportin with a hepcidin-resistant ferroportin. Now, the second step was to try to do this in a tissue-specific way, and we had to do this in a tissue-specific way to avoid the confounded effects of changes in plasma iron availability. And here we used a well-established strategy, and that is to use tissue-specific Cre recombinases. And so over the subsequent 8 years, we went on to try to understand how perturbations in these proteins in a tissue-specific way affected the tissue, not just in terms of iron levels, but also the physiological function of the tissue itself. So let's start with the heart. The first mouse we generated was one harboring a cardiomyocyte-specific deletion of ferroportin. And in this model, essentially, cardiac cells are unable to export iron. What happens in these mice is that they develop fatal heart failure within 3 months. And this was a result of cardiac iron toxicity and buildup of iron in cardiac myocytes. I must also add that this happens despite the fact that plasma iron levels are completely normal. They are unchanged when compared to control mice. So just under control conditions, where plasma iron availability is normal, you have a toxic buildup of iron levels in the cardiomyocyte if you remove their ability to export iron. This is essentially iron overload cardiomyopathy. Now, we thought, well, maybe you can mitigate against this iron overload by reducing the amount of iron available in the plasma. And indeed, when we put the animals on an iron low diet or an iron deficient diet, we were able to protect them from developing heart failure because we balanced iron levels within the cardiac cells by reducing uptake. And this has told us something important. It told us that iron levels within the cardiac cells were the sum of uptake and release. Not surprising. The second mouse we made was one that harbors a cardiomyocyte-specific deletion of hepcidin. And what we found was that these cardiac cells are unable to regulate iron export, and so they're iron leaky. These mice develop heart failure as a consequence of cardiac iron deficiency. The iron-deficient myocardium has a reduction in oxidative phosphorylation and shows a shift towards glycolysis. As before, we thought maybe we can mitigate against this by changing plasma iron availability. And indeed, we can prevent these mice from developing heart failure by replenishing cardiac iron levels through intravenous iron replacement. The third mouse we made in the heart was that of a cardiomyocyte-specific knock-in of the hepcidin-resistant ferroportin. So these cardiomyocytes produce hepcidin, but their hepcidin is unable to inhibit ferroportin cell autonomously. And the phenotype of these mice mirrored that of cardiomyocyte-specific hepcidin deletion, both in terms of the nature of heart failure and its time course. And this really told us that the importance of cardiomyocyte hepcidin to heart function was dependent on its cell autonomous action on cardiomyocyte ferroportin. Now, let's move on to the pulmonary vasculature, specifically pulmonary arterial smooth muscle cells. We generated mice harboring the hepcidin-resistant ferroportin in smooth muscle cells. The Cree transgene we used was not specific to vascular smooth muscle cells. It was for all smooth muscle cells. But interestingly, when we look at these mice, the expression of the hepcidin-resistant ferroportin did not raise ferroportin in all smooth muscle cells. The most dramatic and profound increase in ferroportin expression was seen in smooth muscle cells of the pulmonary arteries. And this led us to think that perhaps we should focus on the pulmonary function in these mice. And when we did that, we noted that these mice developed pulmonary arterial hypertension, leading to right heart failure within 6 weeks. Essentially, pulmonary arterial smooth muscle cells become iron deficient because they are iron leaky. They consequently increase their expression of the vasoconstrictor endothelin 1, and this in turn leads to the development of pulmonary arterial hypertension in these mice. As before, we thought we may be able to prevent this from happening, either by replenishing iron levels in these cells, or, in this case, also interfering with endothelin-1 receptor signaling. And indeed, this was the case. We were able to prevent and partially reverse the development of pulmonary arterial hypertension by treating these mice either with intravenous iron or with an endothelin receptor antagonist. Let's move on to the fetal hepatocyte. Now, it had been assumed for quite a long time that fetal liver hepcidin functions in an endocrine manner to inhibit iron ferroportin in the placenta. So ferroportin is abundant in the placenta, and it was thought that fetal liver hepcidin simply regulates iron supply to the fetal circulation by inhibiting ferroportin in the placenta. This assumption went on and verified for quite a long time, and I decided that I needed to do something about it. I needed formal evidence. And so what we did was we generated mice with a liver-specific deletion of hepcidin. And we used a promoter that we know is activated in fetal hepatocytes in the third trimester. What normally happens in the third trimester in mice and in humans is that there is a rapid buildup of liver iron stores within hepatocytes specifically. And this is required to support subsequent growth that comes after birth. Now, when we deleted liver hepcidin, we noted that the fetuses failed to build up adequate liver iron stores. And furthermore, this failure to build up liver iron stores meant that they were unable to support hepatic hematopoiesis, which occurs obviously in the liver at this stage of development. And as a consequence, there was transient fetal anemia. So this was very surprising to us. It told us that hepatocyte hepcidin in the fetus was involved in the cellular autonomous control of ferroportin to allow the buildup of liver iron stores, and that this buildup in itself was essential to support hepatic hematopoiesis. This is entirely new information we didn't know about. When we generated mice with a liver-specific knock-in of hepcidin-resistant ferroportin, the fetuses had exactly the same phenotype as those lacking hepatocyte hepcidin. Again, confirming this idea that fetal liver hepcidin operates cell autonomously to allow the rapid buildup of liver iron stores. Let's move on to the kidney. Expression of ferroportin in the kidney had been known about for a very long time. In fact, well before I even started in the iron field. The speculation at the time was that ferroportin in the renal tubules was primarily involved in iron reabsorption back into the circulation from urine. Although this was disputed by some because there was uncertainty as to whether ferroportin phased into the apical side or the basal side. Again, I don't like speculation, and so I thought, let's go for formal evidence. Let's interrogate this formally. What we did first was to generate mice harboring a renal tubule-specific deletion of ferroportin. And what we did notice was that there was a decrease in iron reabsorption, resulting in a decrease in plasma iron levels, and consequently also a transient anemia. And this confirmed the long-held speculation that ferroportin in the renal tubules is indeed involved in iron reabsorption. However, the change in iron reabsorption was transient and was in fact quite quickly compensated for by an increase in dietary iron absorption. And this told us that while ferroportin in renal tubules is indeed involved in iron reabsorption, this function does not seem to be essential for the control of systemic iron homeostasis. Instead, the most consistent and persistent phenotype we saw with these mice is that they had renal iron overload. And so the deletion of ferroportin in renal tubules caused retention of iron within the renal tubules and a rapid buildup. And this told us that ferroportin in the renal tubules has a dual function of A, controlling iron levels locally within the renal tubules, and B, controlling iron reabsorption back into the plasma. We then went on to generate mice with a renal tubule-specific knock-in of the hepcidin-resistant ferroportin. And similarly, conversely, what we had was a decrease in iron, an increase in iron reabsorption. But again, this was transient. The most consistent phenotype was renal iron deficiency, again highlighting this dual function of ferroportin and of hepcidin, both in the control of renal iron levels and in the control of iron reabsorption. This dual function came into play particularly when we challenged the animals with an iron-rich diet. Typically, when we challenge the animals with iron-rich diet, we see a systemic increase in iron load within tissues, including the kidney. Now, interestingly, when we challenged these mice with an iron-rich diet, they loaded up with iron more than control mice on the same diet in most tissues except the kidney. In fact, their kidneys were relatively protected from iron overload. And this told us that under iron-rich conditions, inhibition of iron reabsorption can exacerbate renal iron overload while reducing tissue iron overload. And conversely, loss of regulation of renal iron reabsorption protects the kidney from iron overload, while increasing iron overload elsewhere. So after all this, we've learned some important and exciting lessons, in my view. The first lesson is that the local hepcidin ferroportin axis is required for the balancing act of iron homeostasis. So when there is an increase in iron availability, iron export in tissues, or some tissues, can redress the balance by increasing iron flux out of tissues and restoring intracellular iron levels to restore the balance. Conversely, when iron availability is decreased, hepcidin inhibits local iron export to increase cellular iron retention and again restore the balance. And so I see the local hepcidin ferroportin axis as a sophisticated overflow control system, which is designed to maintain adequate intracellular iron levels in face of seesawing changes in iron availability. The second lesson I have personally learned from this, and perhaps most rewarding and surprising, is that iron or cellular iron has fundamental important role in some really fundamental physiological processes we hadn't thought of before. We've shown you that cellular iron is important for the control of vascular tone. Cellular iron in fetal hepatocytes supported hematopoiesis. Cellular iron in renal tubules regulated reabsorption. And that cellular iron in cardiac myocytes regulated energetics. And our collaborators have shown other functions for cellular iron, for instance, in dendritic cells in the context of gut mucosal healing. I'm sure there are many more examples of this that we have to discover and work on. And then within this, there is another exciting question, is to try to understand how does cellular iron regulate these fundamental processes? We are already beginning to gain some insight into this how question. So I showed you earlier that the iron-deficient cardiomyocytes experience a decrease in oxidative phosphorylation. And this is perhaps not surprising given what we know about the important role of iron in mitochondrial enzymes. What perhaps is most surprising is that iron deficient cells appear to mount a pseudo-hypoxic response. So the cardiac cells that are iron deficient increase the expression of glycolytic enzymes. Pulmonary arterial smooth muscle cells that are iron deficient increase the expression of endothelin-1. Endothelin-1 is a hypoxia-regulated gene. Glycolytic enzymes are also hypoxia-regulated. And we think what's happening is that iron deficiency within cells causes a normoxic stabilization of hypoxia-inducible factors. So you'll all know that these hypoxia-inducible factors are essential for orchestrating cellular responses to hypoxia. They are continuously produced in the cell and degraded because of the action of oxygen-requiring polyl hydroxylases, PHDs, and factor-inhibiting FIH. Now, these enzymes also require iron as a cofactor. And so in these iron-deficient cells, these enzymes are inactivated, allowing HIF to be stabilized, translocated to the nucleus, and drive the transcription of genes that would normally be transcribed in response to hypoxia. And I think a lot more work needs to be done to try to understand how cellular iron regulates all these other fundamental physiological processes, because this could be a way for us to try to understand why iron deficiency is such bad news for our organs, not just in terms of causing anemia, but how tissue iron deficiency affects the functions of our organs. Well, this was all very interesting, exciting for me personally and for my team, but we have to ask ourselves the so what question. This is not a purely academic exercise. We have to try to understand the implications of our work for the health of humans. And I think a good way to start is to try to think about the scenarios in which local iron homeostasis might be perturbed. There are many, many scenarios that we know of, many others I'm sure we have yet to discover. Today I will give you 3 examples for which we already have some evidence. The first example relates to the fact that hepcidin is essentially an acute phase protein. Hepcidin has a STAT3 response element, making it induced by interleukin-6, the inflammatory cytokine interleukin-6. This is the product of an evolutionary arms race between host and pathogen for iron. When we have an infection and ensuing inflammation and increase in iron-6, we produce hepcidin to reduce plasma iron availability, to keep iron away from blood-borne pathogens. There is a caveat to this, though. Inflammation in iron-6 isn't just a hallmark of infectious inflammation. It is also a hallmark of modern human diseases, chronic conditions, such as chronic kidney disease, chronic heart failure, cancer, and chronic obstructive pulmonary disease. And now, this now gives us a greater understanding of why iron deficiency is so prevalent in these conditions. What makes this even more interesting is that this iron deficiency, independently of anemia, has recently been recognized as a comorbidity and a driver of adverse outcomes in these patients. Now, this really highlights iron deficiency of chronic disease as a very strategic target. If tackled correctly, there is the potential to benefit large numbers of patients. But if tackled badly, there's the potential to cause enormous harm to large numbers of patients. And as ever, tackling something correctly requires understanding the entity in question. So let's see how our discoveries change our understanding of iron deficiency of chronic disease. So this is a simplistic diagram of iron homeostasis. You've got the tissues that supply iron, mainly the spleen, the liver, and the gut. We have the plasma in pink. And we have iron requiring tissues. Here we've got the pulmonary vasculature, the heart, and the kidney. Now, until recently, the model of iron deficiency of chronic disease was as follows. Inflammation increases hepcidin expression in the liver, and this then blocks iron export out of the liver, the gut, and the spleen, causing a decrease in plasma iron availability, and consequently, over time, also a decrease in iron levels within these peripheral tissues, because there's simply less iron in the plasma to take on. Now, this picture looks quite different when you then realize that the pulmonary vasculature, hepcidin, heart, and kidney also express ferroportin, and in fact, also express their own hepcidin in response to inflammation. The picture indeed becomes very different. And so what you have in iron deficiency chronic disease is a gradual buildup of iron within these tissues as a consequence of the inhibition of local ferroportin, both by locally produced hepcidin and by the endocrine action of liver-derived hepcidin. So what does this mean? What this means is that the way we diagnose iron deficiency is not optimal. We sample the blood, but plasma iron deficiency does not reflect tissue iron needs. And in fact, altered iron status may itself contribute to adverse outcomes, and consequently may itself be the target that we should be looking at. Currently, we don't do that. The second implication is that if you were then to focus on this plasma iron deficiency and try to correct it, let's say with intravenous iron replacement therapy, you run the danger of unintentionally increasing further iron levels within these tissues with the potential for injury. So you have increased plasma iron availability in the face of blocked iron export in these tissues. And this leads us to the third implication, and that perhaps in iron deficiency chronic disease, we should be using an alternative approach to tackling iron deficiency, and that is of normalizing hepcidin levels. And normalizing hepcidin levels would restore both plasma iron availability by unblocking ferroportin in the liver, gut, and spleen, but would also restore the tissue's ability, the heart, kidney, and pulmonary vasculature, their ability to control their own iron levels. Let's move on to the second example. And the second example relates to the fact that hepcidin is also regulated by bone morphogenetic proteins. The main agonists for hepcidin production are BMP6 and BMP2. They signal to the hepcidin promoter via a BMP response element and require for this a BMP receptor complex. Now, quite separately, BMPR2 mutations have been known for a long time to cause, or some mutations in BMPR2 have been known for a long time to cause hereditary pulmonary arterial hypertension. This is when, as I mentioned earlier, there is a remodeling of the pulmonary vasculature due to change in the phenotype of the smooth muscle cells, leading to a rise in pulmonary arterial pressure and sometimes also right heart failure. Now, if you recall correctly in an earlier slide, this is exactly the same phenotype that mice harboring hepcidin-resistant ferroportin in smooth muscle cells develop. If you remember, I told you that pulmonary arterial smooth muscle cells from these cells become iron deficient because they're iron leaky, and that they increase expression of endothelin-1, and that this accounts for their phenotype. Now, this led us to think, well, perhaps the same is happening in the pulmonary arterial smooth muscle cells within patients. And so we were able to acquire some cells, pulmonary arterial smooth muscle cells, from patients carrying these BMPR2 mutations. And to our surprise and delight, we found that these cells were deficient in the production of hepcidin when compared to cells taken from healthy controls. So it appears that BMPR2 mutations impair the production of hepcidin in pulmonary arterial smooth muscle cells. Furthermore, these cells had a greater expression of ferroportin than cells from healthy controls, and also greater expression of endothelin-1. If you recall correctly, we were able to prevent and partially reverse the development of pulmonary arterial hypertension in mice by giving them intravenous iron to replenish iron levels within the pulmonary vasculature, and this decreased endothelin-1 levels as well. In tissue culture, when we take pulmonary arterial smooth muscle cells from patients and we give them either iron or exogenous hepcidin, we were also able to reduce the expression of endothelin-1. And this leads us to think or hypothesize perhaps a similar treatment in carriers of BMPR2 mutations could be used to prevent the development of pulmonary arterial hypertension. Now, a third example, and this relates to the regulation of hepcidin by hypoxia-inducible factor 2. This was very recently described by our collaborators at the University of Michigan. They essentially discovered that hepcidin is, at least in colorectal cancer cells, is regulated by HIF2 alpha, binding to an HRE, hypoxia response element in the hepcidin promoter. Furthermore, they found that hepcidin was dramatically increased in colorectal cancer cells compared to healthy cells, and that this increase in hepcidin was driven by HIF2 alpha in the colorectal cancer cells. They found that this increase in hepcidin completely blocked ferroportin in the tumor cells, causing intracellular iron retention, which was required for supporting cancer nucleotide metabolism and mitochondrial function, and required to promote tumor growth. They've shown this in mice, and they've also been able to show that increased hepcidin production or hepcidin expression in colorectal cancers in patients was associated with tumor growth and worse outcomes. So in response to the so what question, we can say that the local hepcidin ferroportin axis appears to be disrupted in many, many disease processes. Chronic iron deficiency of chronic disease, pulmonary arterial hypertension, cancer. These are the ones I've talked about, and there are many others. And that the ensuing changes in local iron homeostasis as a result of the disruption of the local hepcidin ferroportin axis can then either drive disease, as we think it does in pulmonary arterial hypertension, modify disease, or alter response to treatment, as we think it does in iron deficiency of chronic disease and in cancer. And so all this together brings us to think perhaps there is value in targeting local iron homeostasis, or more specifically, the pathways that are regulated locally by iron levels or cellular iron levels, as an alternative approach to systemic iron intervention. So systemic iron interventions are indiscriminate. They deliver large quantities of iron, whereas perhaps focusing on the pathways that are regulated downstream of local iron deficiency or local iron overload can give us a way into achieving specificity without systemic iron intervention and without the danger of flooding the system with iron levels. On that note, I want to acknowledge everyone who has contributed one way to another, either to this work or to supporting me in my career. I want to thank my lab members, past and present, Goran, Mohammed, who was primarily involved in the kidney work. Mayra Vera, who has recently joined my lab and will be taking the cardiac work further. Lara Camara, who was involved in the placental work. Paul Loic, who was involved in some of the smooth muscle cell work. Eugene Chung, who was involved in some cardiac work, and Magda Wolna, who supported various lab members over the years. I want to thank my sources of funding, most notably the BHF, who awarded me a BHF Intermediate Research Fellowship in 2013, which allowed me to form my lab, and more recently, the MRC, who awarded me a Senior Research Fellowship, which is now allowing me to translate my basic findings into clinically relevant discoveries. I want to thank my collaborators everywhere, here in the UK and abroad, and list is far too long to go over it. But I would like to also take time to thank my mentors, and I've had many over the years. I suspect some of them don't even know that they are my mentors, because I tend to ask for advice and guidance, and I always get it. Primarily Professor Peter Roberts, Professor Dem K. Davies, Peter Ratcliffe, Colin Bagent, and David Patterson. And also thank you all for listening to my talk, and I do hope I've changed your view of ions. Thank you.
Samira, thank you so much for giving that talk. It was a magnificent talk and a wonderful sort of overview of your journey, of your scientific journey. What I really liked about it was how you started at the very beginning. And when I say the very beginning, almost at the very beginning of time. Often, you know, I tell students they should take note of history. And often I'm talking about early work by seminal scientists, but you go way beyond that. So it falls on me. It's my honor to actually, you know, give you this vote of thanks. But actually, if we were in physical times, I would have an award to give to you. So I think that particular honor will belong to your postee, your postman. So I would like to say thank you, and also to just sort of what I really liked about, you know, just maybe pick up on a few things. So what I really liked, you know, you began partly by saying, oh, you know, I discovered this Friday night experiment. I think it's really evocative. And how you discovered something that you thought was a bit strange, a bit curious, and you really started to follow it up, you know, something that was clearly new, and the way you went about it, by developing the appropriate tools, by laying the foundations, the basic principles, which, you know, in a number of different organs and tissues, which has really allowed you to, you know, become, you know, the expert in this field. And by having all those tools, presumably in years to come, you will have, and as you've already presented, amazing collaborations, which will really help you cement your place in the scientific history. And so I think that's you know, with my ECR, my early career hat on, I think that's a real lesson for any early career researcher that is watching this presentation. You know, I think that they can learn a lot from those processes and about how to go about really developing your research identity. And so I really want to say thank you very much. And I'm sure that everybody that is listening would like to say the same. I just want to give a note. We're running a little bit over time. If anyone's got any questions for Samira, traditionally we don't have questions in these prize lectures. So if you do have any questions, she will be at the Meet the Speaker session later. So if you would like to talk to her and ask her anything specific about her work, then you should be able to do so then. We've actually only got 5 minutes, I think, before the next session starts. So with that, I would like to again say, you know, thank you profusely, and you know, hopefully I'll get to meet you in person in future. But yeah, anyone that's watching, enjoy the rest of your day. Thank you very much.